cirrhosis, where the function of the liver is severely compromised, and then it can progress on to there to cancer known as hepatocellular carcinoma or HCC. Now, I would say the most important point that I want to emphasize about MASH is that liver fibrosis in MASH, which is caused by hepatic stellate cell activation, is the major determinant of liver failure and death from other causes. It is also the most difficult feature of MASH to treat. And in fact, some of the headlines that you've seen recently, as recently as earlier this week or late last week, about FXR activators and about incretin therapy. If you look closely at the data and you look at the improvement in fibrosis compared to placebo, where you always get a large placebo effect, it's actually quite modest, even though that makes the headlines. Now, another very interesting point about MASH that many people perhaps do not appreciate is the leading cause of death among MASH subjects is actually not liver failure, but rather atherosclerotic cardiovascular disease. And this is a really poorly understood area. It used to be thought that it was merely due to shared risk factors, but over the last 10 years or so, that has been proven not to be the case, that there's something special about MASH. And most importantly, it's MASH fibrosis that is most associated with atherosclerotic cardiovascular disease. And I won't talk about that today, except at the very end to suggest an important point related to translational relevance. So one of the features, as we know, in MASH is hepatocyte death. And over the years, people have speculated that hepatocyte death is important because it can stimulate inflammation and it can somehow lead to liver fibrosis. And people have suggested that the two major causes of hepatocyte death in MASH liver is necroptosis and apoptosis. And we had to go back and re-examine that, and we agree with that conclusion. But how that could lead to liver fibrosis is really very, there are many, many gaps in how it leads to liver fibrosis. And again, I want to emphasize that when we talk about liver fibrosis, we're talking about the activation of hepatic stellate cells, or HSCs. So throughout my talk, you'll see the abbreviation HSC. And those are the collagen-forming cells that in an activated state lead to liver fibrosis in MASH. So how does this all occur? And we look at the world of cell death, not so much directed as the direct effect of the dead cells themselves, but the fate of those dead cells, what happens to them. And in particular, do they get cleared largely by macrophages or do they not? And that is the topic of efferocytosis, which to a large extent is the most important topic when you're talking about cell death. So macrophages have receptors that recognize dead cells. Here I abbreviate apoptotic cell by AC, but there are certain analogies to necroptotic cells as well. And you'll see AC as an abbreviation that I use throughout apoptotic cell. And these receptors can bind the dead cells and engulf them, and engulfing them gets rid of the dead cells to a large degree. And so in the simplistic scenario, this leads to safe disposal of apoptotic cells to prevent necrosis and inflammation. But even more importantly, when a macrophage takes up a dead cell, to a large extent because of the material delivered to the macrophage from the degradation of that dead cell, amino acids, fats, nucleic acids, etc., other metabolites, that it leads to macrophage reprogramming. And that is another point that I want to emphasize. And the macrophages are reprogrammed into what we call a pro-resolving or a pro-repair state. And among the reprogramming features is turning the macrophage into a factory of mediators of inflammation resolution, for one of the most famous of which, for example, is interleukin 10. Another one happens to be TGF beta, but that's different in the liver. So macrophage reprogramming. Now, a very important aspect of macrophage reprogramming is changing the macrophages into becoming super efferocytes. And what I mean by that is the ability of macrophages to continually take up apoptotic cells. And we call that process continuing efferocytosis. And so that leads to a cycle, what we call the efferocytosis resolution cycle. And the process whereby macrophages are reprogrammed by primary efferocytosis to become good at continuing efferocytosis is extraordinarily important in vivo. If you block that specific process of continuing efferocytosis in vivo, you markedly impair resolution and repair processes. And then finally, defects promote chronic diseases of impaired inflammation resolution, such as autoimmune diseases like lupus and rheumatoid arthritis, chronic lung disease, neurodegenerative disease, and atherosclerosis. And we've spent quite a number of years studying athero. But surprisingly, there was very, very little about MASH. In fact, virtually nothing when Hongzhu began. And the only thing that's come out over the years that Hongzhu has been working on this are some interesting papers about a molecule called TREM2. But even that work, there are a lot of mechanistic gaps. So we were surprised by how little is known about this topic. So the basic question that we set out to ask a number of years ago when Hongzhu first came to the laboratory was, is the clearance of dead hepatocytes by liver macrophages impaired in MASH? For example, like it is in lupus, like it is in atherosclerosis. So we're going to begin. So it turns out that Hongzhu developed two complementary stories, which turn out to be the fact that they're not the same and complementary turns out to have translational relevance, therapeutic relevance. But it just turns out the way it is. And so he began with a story related to those hepatocytes in MASH that happened to die by necroptosis. And so that's the first topic that I'll quickly review. This paper came out a few years ago. I think it was 2022. So we needed to go back and take a look at human MASH, at whether or not necroptotic hepatocytes were really there. There was literature to this extent, but it was limited literature. So being able to say that a cell died by necroptosis is not easy. And honestly speaking, not 100% definitive. So you can just do the best you can do. So we used two markers. One marker was phospho-MLKL, which is a signaling molecule that leads to the type of programmed necrosis that necroptosis represents. And we found in human MASH liver. I think somebody needs to mute. So we found in human MASH liver that there was clearly an elevation of hepatocytes that stained with phospho-MLKL, which was preliminary evidence that necroptotic death was occurring in human MASH. And then we used a complementary marker called RIP3 kinase and also looked at human MASH, normal steatotic and MASH human liver, and found an increase. So we think that the complementary methods are pretty good evidence. And in fact, we can also look at Western blot data looking at phospho-MLKL in human MASH versus normal liver and RIP3 kinase, et cetera. So I think altogether this supports the few papers that were out there before ours that in fact among the ways that hepatocytes die in MASH is through necroptosis. And of course, we need mouse models to be able to test causation. And so we went to a model that Xiaobo Wang, who is the leader, a faculty leader of the MASH program in my laboratory, um, discovered a model that he discovered in 2016, which is now widely used because of its relevance to human MASH and because it encompasses cardiometabolic risk factors, which is important because that's why they changed the name from NASH to MASH. So we call this the fructose palmitate cholesterol model or FPC model. And again, it's now used by many laboratories. And so we ask the question, does it act like humans with regard to necroptotic hepatocytes? And this is using the marker phospho-MLKL by immunohistochemistry and RIP3 by Western blot, and the answer was yes. So it's mimicking what we see in humans, like many features of this model mimics humans. So we had a hypothesis that during the transition from MASL to MASH, that you get a decrease in hepatocyte necroptosis, and that for some reason there is defective clearance by liver macrophages, and by some mechanism, this activates hepatic stellate cells to lead to liver fibrosis. So just a speculative hypothesis. So the first thing we did is we went into our mouse model and we asked the question, can we find evidence that these necroptotic hepatocytes, in this case marked by RIP3, are poorly engulfed by macrophages, which are green? Now, normally, if you do immunofluorescence like this and you find good engulfment, either the red will really be inside the green macrophage, or depending upon the optics that you present, it will be yellowish. But you can see here that the macrophages are contacting the necroptotic hepatocytes, but not really engulfing them very well and not degrading them because the necroptotic hepatocytes are still quite big. So the strategy that we use to test our hypothesis that this could be important in HSC activation, liver fibrosis, is we first had to determine why the clearance was bad. Now, that in its own right is an important question, but we needed to do that if for no other reason, because that would suggest tools that we could target to test causation. So that was the idea. So we landed on exploring this very classic don't eat me axis in the field of phagocytosis and efferocytosis called the CD47 SERP alpha axis. And basically, we think we've evolved this to make sure that our phagocytes don't eat living cells. So living cells present typically a cell surface molecule called CD47, and that interacts with a macrophage molecule called SERP alpha, which is what we call a don't eat me signal. And when SERP alpha gets activated, it sets up a series of signaling reactions that paralyzes the cytoskeleton and thereby inhibits the engulfment of the dead cell, thereby sparing a living cell from getting inappropriately eaten. Now, the reason why we became interested on this was by a paper in another laboratory. Ironically, the paper's first author was somebody who would eventually come to my laboratory to work on atherosclerosis. And the senior author was a former postdoctoral fellow in my lab, Gabby Fredman. But this paper is Gabby and Brennan Gerlach's independent paper that I had nothing to do with. They were exploring the general issue of engulfment of necroptotic cells. And the first observation they made is that necroptotic cells, and I believe they were just using necroptotic macrophages, and they found that it inappropriately overexpressed CD47, had a high expression of CD47. Normally, nature would want these cells eaten, so it's inappropriate that they have a high expression of the don't eat me molecule CD47. Incidentally, apoptotic cells are appropriate. That's AC. It goes down. But necroptotic cells had this very surprising increase in a don't eat me signal, which would appear to be maladaptive and not what evolution intended. Most importantly, what they found is that under normal conditions on the left, necroptotic cells, the green, were not eaten well by macrophages. But if you neutralize the don't eat me signal, now the macrophages started to eat pieces of the necroptotic cell. So this very basic work by Brennan Gerlach and Gabby Fredman got us to think about the relevance of this to what we observed was poor clearance of necroptotic hepatocytes by liver macrophages in MASH. So we asked, is there an increase in CD47 in MASH? And we began with just a general Western blot. It doesn't say anything about cell type, but it was quite dramatic that CD47 went up in MASH. And then we actually did immunofluorescence microscopy and to ask the question, does CD47 go up in necroptotic hepatocytes? And it did quite dramatically. So that really got us interested in the hypothesis that this was an important mechanism. And then we conducted an ex vivo experiment that was patterned after what Brennan and Gabby did, but this time using necroptotic hepatocytes and macrophages. On the left, as expected, the necroptotic hepatocytes did not get engulfed well. On the right, when we added anti-CD47, we saw very nice engulfment. So the principle that Brennan Gerlach and Gabby Fredman established in their system seems to be relevant to necroptotic hepatocytes in MASH. And again, the arrowheads on the right are pointing to pieces of necroptotic hepatocytes that have been successfully engulfed by the macrophages in the face of anti-CD47. Now, interestingly, this increase in engulfment by anti-CD47 with necroptotic hepatocytes, which you're seeing in the first two bars, is not seen with apoptotic hepatocytes. And so we don't know why that is, but it is what it is. And it was the first clue that apoptotic hepatocytes are going to be a new story. And I'll talk about that in a little bit. So then we went in vivo. And we took this very nice fructose palmitate cholesterol diet model, let them go 8 weeks on the diet, which gives you steatosis. We always like to intervene after steatosis for two reasons. Number one, because that allows us to focus on the all-important transition from steatosis to fibrosis without worrying about effects on steatosis. And number two, if this ever turned into a treatment idea, that's basically the way it would be done in humans. So we let them go 8 weeks. The mice become steatotic. We then treat them with IgG or anti-CD47. We let them go another 8 weeks, which is where the control mice will develop early liver fibrosis. So the first thing we looked at is the ratio of macrophage internalized to total RIP3 positive cells. That's a marker of engulfment. And as predicted from the ex vivo data, it did go up with anti-CD47 treatment. And you would imagine that would lead to more necroptotic cells floating around the liver, to a decrease in necroptotic cells floating around the liver because you are helping to get rid of them. And so the total RIP3 cells go down with anti-CD47. But ultimately, what we care about is liver fibrosis. So did this do anything to liver fibrosis? And so I'm showing you two pieces of many data, Sirius red, a collagen stain, aniline blue, another collagen stain. And in both cases, we got a substantial decrease in liver fibrosis with the anti-CD47 treatment. We also got a decrease in activated hepatic stellate cells marked by alpha SMA, and we got a decrease in gene markers of activated hepatic stellate cells as indicated. We also conducted RNA-seq, and the RNA-seq data indicated a broad decrease in collagen-producing cells or collagen-producing genes in the anti-CD47 treated mice. We also obtained similar data using a different model called the high-fat CDAA, choline deficient amino acid defined steatohepatitis model. It's another good model, but very different. So it's applicable across models. And we also found the same data if we specifically silenced CD47 in hepatocytes instead of using a systemic antibody. And so, as I mentioned before, it takes two to tango in this CD47 SIRP alpha axis. CD47 interacts with SIRP alpha. Now, the fact that CD47 goes up in MASH, in necroptotic hepatocytes, would be all that you would need to mess things up. But we were surprised that macrophage SIRP alpha also went up. So that's a perfect storm. Two bad things happening at once. CD47 on the dead cell going up and SIRP alpha on the macrophage going up. So that was surprising, but very interesting to us. And we went to our experimental model. So this was human. And then we went to our experimental models, both the FPC model and the other model I mentioned, and macrophage SIRP alpha in MASH goes up. And similarly to the ex vivo we did with anti-CD47, we conducted an experiment with anti-SIRP alpha, and we showed that the macrophages now engulf more of the necroptotic hepatocytes when you neutralize anti-SIRP alpha. And when we went in vivo, we found that, when we went in vivo with either of the models, we found that we can improve efferocytosis or engulfment of necroptotic hepatocytes with anti-SIRP alpha injection into the mice. And again, whether we use the FPC model or the other model. And most importantly, we find that anti-SIRP alpha, like anti-CD47, decreases fibrosis, again, in either model, whether you look at Sirius red or the genes. Now, we also conducted recently, or Hong Zhu conducted an experiment recently with a macrophage-specific knockout of SIRP alpha instead of systemic anti-SIRP alpha antibody injection and found the same data. So now the million-dollar question became mechanism. And I'll go into a little bit of, very little bit of unpublished data. This is something that Hong Zhu is going to work on. And it harkens back to a project that Xiaobo Wang in my laboratory published in 2016, where he identified a new mechanism of MASH fibrosis that at the time was completely unrelated to anything having to do with hepatocyte death. In fact, This was a pathway in overtly living macrophages in MASH, living hepatocytes in MASH. And he discovered that a gene regulator called TAZ upregulated a molecule called Indian hedgehog, or IHH, which gets secreted from the hepatocytes, acts on a cognate receptor on hepatic stellate cells, and activates those hepatic stellate cells to become profibrotic. And over the years, Xiaobo has shown backwards and forwards importance in experimental MASH, in human MASH, in translational therapeutic applications, in hepatic cellular carcinoma, in many different aspects. So, and now many labs have looked into this and have reproduced all of our findings and have taken it further. And so, whenever we find an effect on lowering fibrosis, we always ask, could it somehow affect TAZ? And remarkably, we found that when we promote necroptotic hepatocyte clearance, in this case with anti-SERP alpha, this is an in vivo experiment in our FPC model, we markedly lower TAZ. So this is really a dramatic result. And this is all unpublished. And obviously, we're extraordinarily interested in the mechanism of this effect. And does this account for the anti-fibrotic effect of TAZ? And we already have in vivo data suggest that it does. So really the million-dollar question is, how does improving the clearance of necroptotic hepatocytes, how does that lower TAZ in other living hepatocytes? And it obviously involves cell crosstalk between the engulfing macrophages and the living hepatocytes, but that's all future work. So the hypothesis and therapeutic implications is that in MASH, there is a defect in the CD47 SERP alpha axis leading to HSC activation and liver fibrosis. And if we can neutralize or silence hepatocyte CD47 or macrophage SERP alpha, will restore the engulfment of the necroptotic hepatocytes, lower TAZ in hepatocytes, and lead to HSC deactivation and resolve liver fibrosis. Okay, we're going to move on to the final topic, which is apoptotic hepatocytes. And this is, as of this day, unpublished, but we expect that it will be coming out very, very soon. So again, what is the fate of apoptotic hepatocytes in MASH? So this is a topic about which virtually nothing was known. Again, I did mention that in the course of this work, a TREM2 paper came out. TREM2 is an efferocytosis receptor, but the sort of robust evaluation of efferocytosis in vivo is something that was a major gap even up to this point. So the first thing we did is go to humans and ask the question, if you look at early human MASLD versus advanced MASH, do you find a defect in the uptake or efferocytosis of apoptotic macrophages? And so again, we have this metric of ratio of macrophage associated to free dead cells. In this case, it's tunnel. We also use cleave caspase 3 as a more specific marker of apoptosis, and the data are basically the same. But you can see in human MASH that efferocytosis, which is the left graph, goes down, and the number of uncleared dead cells goes up. So clearance of apoptotic hepatocytes is effective in MASH. And it's also defective in our experimental models. And in this case, I'm showing you the high-fat CDAA model, showing a progressive decrease in FRO on the left and a progressive increase in the accumulation of apoptotic hepatocytes on the right. And here we're using a more specific marker of apoptotic cells cleaved to caspase-3. Again, project design exactly as before. First, determine the mechanism, A, because that's important, but B, because it will give us tools to be able to manipulate the mice to be able to determine if it has anything to do with liver fibrosis. So in this case, we, knowing from the previous data that you may remember my showing you, that CD47 SIRP alpha story did not apply to apoptotic hepatocytes, we knew it had to be something different. And we focused on a very famous marker of the resident liver macrophages called Kupffer cells, TIM4. Now, most people who study Kupffer cells use TIM4 as an excellent marker of KCs. But it turns out from work years ago by Shikanagawa that TIM4 is a bona fide efferocytosis receptor. And we became interested on this because Chris Glass and now others showed, at least at an mRNA level, that TIM4 mRNA, where it's very highly expressed on Kupffer cells in normal liver, is not well expressed at all, virtually undetectable, on MASH macrophages, which are macrophages that come in from blood-borne monocytes, occurring as the resident Kupffer cells slowly die away. And so, again, we go right to human, and we ask the question, does TIM4 protein go down on human MASH macrophages? Remember, the early work by Chris Glass was mRNA data in mice. Now we're looking at protein data in humans. So an important leap. And in fact, it does go down. TIM4 expression by MASH macrophages is lower compared to Kupffer cell macrophages in normal liver in humans. And in our experimental model, this is also the case, looking at TIM4 MFI mean fluorescence intensity in Kupffer cells. Those are the CLEC4F, another marker, is just a long way of saying that it goes down in Kupffer cells. So the cause, now we had a tool. We knew the mechanism and we had a tool. So we could knock down TIM4 in Kupffer cells using a CLEC4 FCRE and ask the question, does this accelerate the progression to MASH? Now, TIM4 would go down normally as MASH progresses anyway, so it would make zero sense to knock down TIM4 after MASH develops because it's not there anyway. But the idea here was to knock it down when it's still there and ask, does this accelerate MASH? So we got good knockdown, shown in this graph, and we got a nice decrease in efferocytosis as predicted, and we got a acceleration of liver fibrosis. In this case, I'm showing you serious red data and collagen 1A1 area data. But the most important experiment that Hongzhu did was to put back TIM4 in MASH. And he did that by creating an inducible TIM4 mouse. I will not have time to go into the design, but it's a classic design in which you use doxycycline and a cell-specific inducer model, in this case, a macrophage inducer called TTA, and doxycycline will, at the end of this series of reactions, cause the gene of interest to go up in your cell type of interest, in this case, macrophages. So we call this an inducible transgenic, or ITG. And he very successfully put back TIM4 in MASH macrophages. The doxycycline was added after MASH developed, where TIM4 would normally be very low. You can see in the upper image very little red. And so this strategy worked very nicely in order to restore TIM4 to MASH liver, i.e., in a setting where TIM4 is normally very low. And of course, the idea would be, can we make things better by restoring TIM4? And so we looked at efferocytosis, and it was improved. And we looked at fibrosis, and it was improved, namely, it went down. So again, we were very satisfied with these data, but we needed to understand the mechanism. Why would improving the uptake of apoptotic hepatocytes somehow decrease fibrogenesis in MASH? And again, I'll remind you that one of the most important features of efferocytosis is macrophage reprogramming stimulated by the uptake of a dead cell that leads to various processes of inflammation resolution and tissue repair, not to mention continuing efferocytosis and the efferocytosis resolution cycle. So Hongzhu, with great intuition, wanted to focus on one known mediator of inflammation resolution, interleukin-10. We were quite familiar with this particular mediator from our work in atherosclerosis, where efferocytosis is also defective, where stimulating efferocytosis makes things better, and where macrophage reprogramming to make more IL-10 in atherosclerosis is an important part of the story. But Hongzhu was interested in this in MASH because of a number of papers, some of the references I put in the lower left, indicating that interleukin-10 may have anti-fibrotic roles in the liver. So in preliminary data, what Hongzhu showed is that incubation of Kupffer cells that have good TIM4 and are good efferocytes with apoptotic hepatocytes induces IL-10. So KCs eating hepatocytes is similar to macrophages eating apoptotic Jurkat cells, which is our typical model that we use. He also found that interleukin-10 from these efferocytosing Kupffer cells, in other words, conditioned media, could deactivate the hepatic stellate cells. Conditioned media plus or minus neutralizing IL-10 antibody. And finally, he went in vivo and found that liver IL-10 is increased by restoration of macrophage TIM4. In other words, by that inducible transgenic model. So these three pieces of data were strong evidence in support of his idea. But to test causation, what Hongzhu did was to try to neutralize IL-10 when we improve efferocytosis in our inducible model and ask whether that ruins the improvement induced by restoration of TIM4. And what he found, if you focus first on the black bars, that is basically reproducing what we did with the inducible TIM4 transgenic. So just focus on the black bars for the time being. We see an improvement in serious red fibrosis and other markers of HSC activation. So those are the black bars, bars number 1 and 3. The red bars are what we see when we put in the anti-IL-10 receptor. And so I want you to focus on bars 3 and 4 in each graph. You can see that the benefit of the inducible transgenic became abrogated. The fibrosis was no longer improved. The decrease in collagen 1A1 was no longer down, et cetera, et cetera. So this provided in vivo causation evidence that interleukin 10 was playing an important role in the benefit conferred by restoring macrophage TIM4 in MASH and by restoring efferocytosis in MASH. So again, hypothesis and therapeutic implications. In MASH, because TIM4 goes down in macrophages, you have defective uptake of apoptotic hepatocytes, and that leads to activated hepatic stellate cells and liver fibrosis. We could put back macrophage TIM4. Now, we think that this can be done therapeutically. We did it genetically with the inducible TIM4. But we think that there are therapeutic modalities. For example, a very hot topic is designer macrophages or CAR-M. And we have data to indicate that that can do the same thing that the inducible transgenic can do. Uh, Lipid nanoparticle mRNA therapy evolving from COVID as a modality of high interest for humans now. We have evidence that that can do it as well. So we believe that there are modalities relevant to humans that can restore TIM4 and that induces IL-10 and maybe other anti-fibrotic factors, and again, deactivates hepatic stellate cells and mitigates liver fibrosis. I'll show you a little bit about the macrophage therapy. This was done in collaboration with Mike Klaczynski and colleagues at Charisma Therapeutics, where hematopoietic stem cells from mice were transduced with TIMD4 control, differentiated into macrophages, and injected into MASH mice. And on the Western blot, you can see that we did a good job of restoring TIM4 in macrophages in the mice that received the TIMD4 macrophage therapy compared to the mice that received the control macrophages. We then looked in the liver of these MASH mice. This happened to be the HFCDA model, and we got an improvement of efferocytosis, and we got a very nice decrease in liver fibrosis compared to the macrophages that got control macrophages. And just by way of comparison, we're showing you normal non-MASH liver on the far left. So I'm going to end with the concept of integrated macrophage therapy. So as nature dealt us the clearance of necroptotic hepatocytes and apoptotic hepatocytes lead to complementary effects in terms of deactivating hepatic stellate cells. One we think involves the TAS-IHH pathway, and one we think involves IL-10. And so those are different and they're complementary. So if we can restore TIM4 and we can also silence macrophage SERP alpha, both of which can be done incidentally with lipid nanoparticle RNA therapy, TIMD4 plus SI-SERP alpha, then we think we can get at least an additive benefit. Now, I will end with reminding you that the leading cause of death in MASH is not liver disease, but atherosclerotic vascular disease. And so the last slide I'm going to show you is to imagine this dual MASH therapy now applying to the number one cause of death in these people, atherosclerosis. Because in fact, silencing SERP alpha has already been shown by Nick Leeper to improve atherosclerosis. And we believe that overexpressing TIM4 in atherosclerotic lesional macrophages will improve the defective efferocytosis that we know occurs in athero. And to the extent that these are systemic therapies, we would imagine that these MASH treatments will also improve atherosclerosis. And in fact, we think it might be a dual improvement because not only will improving efferocytosis in athero directly make athero better, but since we know there's a link between fibrotic MASH and athero, by improving fibrotic MASH, we think that that will secondarily improve atherosclerosis. So atherosclerosis, in theory, would be improved by both a direct mechanism of improving efferocytosis and a secondary mechanism by decreasing MASH fibrosis. So that is how I would like to end. And again, Hangzhe commandeered this series of studies. Xiaobo is the faculty leader of the MASH program in my lab and basically teaches everybody everything and runs everything and teaches me. And then I list a lot of our collaborators over the last several years on these projects and our funding sources. So again, thank you so much for this invitation, and I will be very happy to take questions.
Thank you, Aaron, for the great talk about efferoptosis and how does the macrophage efferoptosis control the MASH progression. So now I move let my partner Julie please take over.
Thank you, Ara, for a fascinating talk, and really learned a lot. And now we will go to the panelist discussion first, and followed by the audience Q&A question part. Hopefully, we finish by time. I know that at 9 o'clock you will have another meeting. So our first panelist will be Dr. Hongyuan Robert Yang. Hi, Dr. Yang. You can just show your video and I'm going to say hello. And I'm just give a brief introduction of Dr. Yang. Dr. Yang is currently a professor in the Department of Integrative Biology and Pharmacology at UT Houston, the same institute as me. And Dr. Yang is an internationally renowned expert in lipid metabolism. And his lab is mainly focused on uncover how cellular lipids are sorted, transported in health and disease. He have identified key regulators of many key proteins that controls lipid droplet growth, lipid composition, lipid delivery, and